Hello boys, welcome back to another interesting session of video tutorials, and this session is meant for class 12th biology students, and this is meant for 25th August 2020. As usual, our memory helps us in understanding what we have been studying in the past and try to connect the past to the present. If you recollect from your memory, we were talking about replication of DNA and various experiments that proved beyond doubt that DNA is the genetic material, and we were also talking about what is called central dogma. And in the central dogma there are basically two steps. One is transcription and translation, and major part of transcription process is handled by mRNA. So we were talking about the structure of messenger RNA. So it's RNA world, we can say. And there are basically three types of RNAs. One is mRNA, messenger RNA. We talked about the role of mRNA in transcription, how mRNA gets the genetic secret messages in the form of code language from DNA to RNA. Since it is carrying messages, we can call that RNA as mRNA, messenger RNA. Similarly, the second one is rRNA, as you are saying, ribosomal RNA. And you can see the structure. Basically, the ribosomal RNA has got two structures, one large subunit and one small subunit. And there is a central protuberance. So something like this is protuberance, central protuberance. So this is how a ribosome will be there, and the RNA that is concerned with ribosomes is ribosomal RNA. So it helps in binding of ribosomes to mRNA. That is the main function of rRNA. It acts as an enzyme, ribozyme, helps in formation of peptide bond during protein synthesis. So one amino acid links with another amino acid because of a bonding, and that bonding is peptide bond. Through peptide bond, proteins will be formed. So basically, RNA was the first genetic material. There is now enough evidence to suggest that essential life process such as metabolism, translation, splicing are evolved around RNA. RNA used to act as genetic material as well as catalyst, like ribozyme we said, catalyst. RNA being a catalyst was reactive and hence unstable. This is an unstable compound. So DNA has evolved from RNA with chemical modifications that make it more stable. So DNA being double-stranded and having complementary strand further resists changes by evolving a process of repair. So on the basis of molecular size and function, three main forms of non-genetic RNA are, as I said, mRNA, rRNA, and one more is tRNA. mRNA constitutes about 3.5% of cellular RNA, tRNA is about 15%, and rRNA is maximum with 80% of the RNA it constitutes. So mRNA or informational RNA or template RNA is a molecule of RNA that helps in transcription from a gene and then translated by ribosomes in order to manufacture proteins. tRNA, on the other hand, is transfer RNA. It is also known as soluble RNA or adapter RNA, which consists of amino acid binding site, which lies opposite to anticodon site. And then rRNA, ribosomal RNA, is a component of the ribosomes, the protein synthetic factories in the cell. It is formed in nucleolus. Again, you must remember this in your competitive exams. rRNA is formed in the nucleolus, not nucleus, nucleolus. So remember this for your NEET exam. And then the basic differences between DNA and RNA. What are those? DNA, of course, was high molecular weight. RNA has very low molecular weight. It's a double-stranded polynucleotide chain with a double helix, thanks to Watson, Professor Watson and Crick. single-stranded polynucleotide chain. Already I said what is polynucleotide and what is a nucleotide, what is a nucleoside. Okay. Deoxyribose sugar is the pentose sugar. Penta means 5, it's a ringed chain, and ribose sugar is the pentose sugar. Only difference is deoxy, one oxygen is less in this. And here nitrogenous base is T, thymine is present. In place of thymine in RNA, uracil is present. And unusual nitrogen bases are absent, unusual nitrogen bases are present. Genetic material of living organisms. If someone asks you what is your secret, we say always my DNA is my secret. And genetic material in some viruses. That's how COVID-19 is doing damage. So viruses have got their genetic material as RNA. RNA polymerase is also called DNA dependent RNA polymerase. DNA because it is depending on DNA. And RNA polymerase 1 is found in nucleolus, again neat exam, and helps in synthesis of ribosomal RNA. Hence nucleolus is called ribosome factories of cell. Remember this, nucleolus, but you not nucleus. Nucleolus is called ribosome factories of cell. And RNA polymerase 2 helps in synthesis of mRNA. Now all these three sentences are golden sentences. You must remember for your competitive exams. RNA polymerase 1 is found in nucleolus, helps in synthesis of rRNA. RNA polymerase 2 helps in the synthesis of mRNA, that plays crucial role in transcription. RNA polymerase 3 helps in synthesis of tRNA. So without this RNA polymerase 1, 2, 3, we cannot exist. There is no secrecy. So thanks to RNA polymerase. So ribosomal RNA is the insoluble RNA. And there is something called Pribnow box. Pribnow box, again, many competitive exams it is asked. It's a DNA sequence found in the promoter region of genes in prokaryotes. It's in prokaryotes. It has the sequence of Tata ATG, ATG. T means thymine, A means adenine, thymine, guanine. And what is Tata box? Not your TV box, of course. Tata box, that's Goldberg-Hagen's box it's called, just like Pribnow box, it's Goldberg box it is referred. It's a DNA sequence found in the promoter region of genes in eukaryotes, that's prokaryotes. Pribnow box is in prokaryotes, Tata box is in eukaryotes. And there it is Tata ATG, here it is Tata Arch. This is the sequence. So keep remembering this, Pribnow box and Tata box. And then we come to, if someone asks you what is the gene, concept of gene. So we say gene is the basic unit of inheritance that expresses specific character. It's the basic unit of inheritance. This is what you should remember. And thanks to Gregor John Mendel, who put the seed of concept called gene. As we studied in our previous chapter, he conducted many experiments with the pea plants, Pisum sativum, remember? And there he did not, or he could not call it as gene, he called it as factors. There are some factors which are responsible for downloading of the characters or traits, said, from parents to offsprings. Now in 1909, Danish botanist Johnson, that's why we have got even in our biology lab, you must have seen microscopes named Johnson and Johnson, okay, proposed that each portion of a chromosome that controls a phenotype is called gene. So this is phenotype, of course, physical appearance of an individual, right? And in Greek, what is gene? To give birth to. In 1941, George Beadle and Edward Tatum, it's called Beadle and Tatum, discovered that genes control the production of enzymes. And they proposed one gene is responsible for one enzyme synthesis. One gene, one enzyme hypothesis. So out of all this, you must remember this for your NEET exam, Johnson. And in 1944, already we discussed, Oswald Avery announced that DNA is the substance responsible for hereditary. And in 1950, Watson and Crick discovered chemical structure. Of course, these professors will not leave you as long as you are a biology student. Many questions will be based on Professor Watson and Crick. And the gene is made up of a specific sequence of DNA nucleotides. We said polynucleotidal chain. And Sam Abenger proposed modern concept of gene while working on E. coli and T4 bacteriophage. What is this modern concept of gene? According to him, gene consists of 4 regions. One is cistron, recon, muton and replicon. These are the things. So what is a cistron? Is the structural gene that is functional. It is functional unit of DNA molecule that codes for specific protein. So you must remember this for not only CBSE, for all your exams. Cistron is a structural gene. It is functional unit of DNA molecule that codes for specific protein. What about recon? It is the unit of DNA that undergoes recombination. That's why recon, recombination. What about mutant? As the name suggests, mutation. It is related to mutation. Mutation is sudden change. It is a unit of DNA that is capable to undergo sudden changes. And again, this gives a clue. Replicon, replication. It is a unit of DNA that undergoes replication. So basically, a gene can be a cistron, a recon, a muton and replicon. This is what it is. And we have got what is called code language, and that genetic code. The 3 specific nucleotide sequence that codes for specific amino acid present in DNA is called genetic code. This is very, very important. Now we are entering into higher level of molecular biology at class 12th. The 3 specific nucleotide sequences. 3 specific nucleotide sequence. Like we said yesterday, initiating codon. Like August I said. Remember, we are in the month of August and this video is meant for 25th August I said. So specific sequence is the codon, triplet codon, AUG, initiating codon, isn't it? So the code is in a specific nucleotide sequence. Now, the 3 specific nucleotide sequence that codes for specific amino acid present in mRNA is called codon. So these 3 nitrogen bases form a codon and they code for a specific amino acid. That I will tell you in a moment in the form of a table. And what is anticodon? The one that decodes the codon. So the 3 specific nucleotide sequence that codes for specific amino acid present in anticodon arm of tRNA. Just now we talked about the tRNA, and while talking we said that tRNA stands for transfer RNA, or it is also called adapter RNA. It consists of amino acid binding site, and this amino acid binding site lies opposite to anticodon site. Okay, and anticodons are complementary codons. So remember when we were talking about complementarity, always Sunil Gavaskar, Ravi Shastri, they used to comment whenever there's a match, particularly when Jawgal Srinath and Venkatesh Prasad were bowling for India. Jawgal Srinath used to bowl half swingers and Venkatesh Prasad used to bowl in swingers. Then we say that both the pace bowlers of India are complementing each other. Similarly, many fast bowlers in the country, the captain will select 2 pace bowlers who will be bowling complementing each other. One in swing, one out swing, or one what we call spin and pace, like that. So that is complementarity. So the general features of genetic code, what are that? Genetic code is a triplet, as we said just now, AUG. Each codon is made up of 3 specific nucleotides. And the genetic code is universal. That is the greatness of this. It is universal for all living organisms, same. Each codon codes for specific amino acid in all living organisms. And genetic codons will not overlap, no overlapping. So adjacent codon never shares their nucleotides. And genetic code is commaless. In grammar, in English, you put a comma to take a break. Here, no break, no without break. Like sometimes you are seeing movies, no, without any commercial break. Like that, here, no break. There is no gap between neighboring codons. And the genetic codon is degenerative in nature. Degenerative means it won't die. More than one codon codes for single amino acid. For example, you can see degeneration. GUA, GUG, GUC, GUE codes for only one amino acid. That amino acid is called valine. Now we come to the triplet code and specific amino acid for which they code. And again, it is very easy to write. Like you have this, just like in genetics chapter, you wrote monohybrid and dihybrid crosses, isn't it? Similar way, you can write UCAG, and here also UCAG. Okay, and here also you can see UCAG, and in this box also UCAG, UCAG, UCAG. Okay, and now you see this U you are picking up, and this U and this U. So U. Okay, then U and C from this side, extreme right. So UUC, UUA, and UUG. This one you are picking up. So that means you are having a triplet code in this fashion. And first two, UUU and UUC, they are responsible for coding a specific amino acid called phenylalanine. Leucine next, like that. Serine, tyrosine, cysteine. And in this stop codons also we are identifying. What is that? UAA, UAG, and UGA. Red letters are there. No, these are stop codons or nonsense codons. You must remember this. Similarly, you must remember this August. And this initiating codon is meant for a specific amino acid called MET, and that is methionine. So this I want you to write right now. Take a paper, pencil, and write this so that you should remember this table. This is genetic code. And further, I will explain to you how translation takes place. And before that, we must understand the structure and role of another RNA called transfer RNA, tRNA. In our next session, we'll take up that. Until